friends, welcome to Tarun IS. Today we will be discussing editorial analysis from the Hindu and the Indian Express of 27th of October 2022. Today's editorial is about the approval given to the genetically modified mustard crop by the GEAC. So we will talk about what are these genetically modified crops, what this GEAC, what is the function of GEAC, and what are the reasons because of which or due to which there have been many protests against the introduction or the commercial trials of the genetically modified crops. And Dhara mustard, which is the hybrid mustard which has been recently given approval, has taken a long years to be given this approval of field testing and seed production. Now if I talk about my personal experience with this, so as a zoology honors student, I had attended one seminar which was delivered by the Deepak Paintel, who is actually the person behind developing this hybrid variety, and he had been the former vice chancellor of University of Delhi too. So he talked about that how he had and his team, along with his team, developed this Buster Barnes system and what this hybrid mustard variety is. So we will talk a little about this mustard variety, and we will also talk about the concerns which have been raised, and then we will talk about that why it is difficult to develop a hybrid mustard variety. So I have taken this article from the Indian Express explained page. So we will see what has been explained about this, and you can relate this article with General Studies paper 3, that is Agriculture, the Cropping Pattern and Application of Science and Technology. Alright, so the context of the news is that the GEC, which is Genetic Engineering Appraisal Committee under the Ministry of Environment, Forest and Climate Change, has given the approval for the seed production and conduct of field demonstration studies. Basically recommended the environmental release of the transgenic hybrid mustard, which is DMH-11. Alright, now this Genetic Engineering Appraisal Committee is the principal body, an autonomous organization under the ministry, to decide that which genetically modified crop is to be given the permission and which genetically modified crop is not to be given the permission. Alright, now basically we have to understand that so far in India only the BT cotton has the approval of the commercial farming. The BT brinjal has faced many protests from the environmentalists and the farmers and the green activists. DMH-11, the hybrid mustard, has after a very long time, because I attended that seminar probably in the year 2013, and I think it has been more than 10 years. It has been like approximately 10 years. After such a long time, this has been given the approval. The environmental release approval has been given. So let's talk about what is this hybrid mustard. So this is the DMHS mustard. This is the DMH-11 mustard. So let's talk about this mustard variety. Alright, now first important thing that you must understand is mustard is a self-pollinating plant. It's a self-pollinating plant. Alright, so you need to remember this, that is a self-pollinating plant. Alright, now we will talk about the hybridization. Now what this hybridization means? Hybrid. That means it consists of two things. Either it is going to have genes from two different varieties, or it is going to have genes from two dissimilar species or different varieties of the same species. But basically, all in all, in conclusion, the essence is that it is going to be two different plants who are going to be produced to one plant with certain inherited characters which are desired, the certain desired traits or certain desired inherited characters. hybridization cannot take place among those traits or characters which are not inherited, right? If there is certain acquired thing and something which is not inherited, hybridization won't work. So the characters or the traits which are required or which are desired should have the heritable feature. That means they should be inherited from one progeny to the another progeny. The principle, the credit for this hybridization technique goes to the an Austrian botanist called by the name Gregor Mendel. And all the botanists and zoologists, they must have studied about the Gregor Mendel. And the credit for the hybridization technique and studying the progeny, the first generation, the second generation progeny in the pea plant, goes to Gregor Mendel. So what is this F generation? The F word stands for the filial. All right, the first filial generation. Now what happens if I consider, let's see, this is a plant. All right, so these are two different mustard plant. Let's say I have taken two different varieties. Now mustard is a self-pollinating plant. So a plant which is self-pollinating needs to, in order for it to undergo the hybridization, I must make it as dimorphic, right? That means it should be able to cross-pollinate. Now this is the process by which I can produce hybrid. All right, taking genes from here, and then producing certain plants. Now these two are the parent crop, and the progeny will be the F1 generation, the first filial generation after the crossing. So we will understand how this hybridization took place and what is the Buster-Barnes system which has been designed, and why this DMH mustard is important. We will talk about that in subsequent slides. So basically, like I said, that hybridization isn't easy in mustard because it is self-pollinating. All right. So this is the picture which I have taken, and you can understand. Brassica is basically a family to which varieties of mustards belong to. All right. So this is basically called as the floral diagram. Those belonging to botany and zoology, they would understand it, but not you don't have to go into the details of this. But this is basically a floral diagram which talks about that how many stamens are there, how many sepals are there. So if this is the flower, so the flower arrangement is drawn in this way. So this is basically the floral diagram, and the arrangement of petals, sepals, anthers, and stigma, etc. It is shown here. So if you look at this, the anther, what is this anther? Anther basically consists of a filament, and on top of it there is a ovule, there is egg-like structure like this. Now this anther, it basically produces what is known as the pollen. Now sometimes when you go to a garden, you see yellow spots on flowers. Now what are these? These are basically the pollens. Pollens are the male gamete. So if you look at here, this is stigma. All right, this is stigma and this is style. So stigma is, it looks something like this. If you look at any flower, you will have in the center a portion which is a little sticky. So that sticky portion is known as the stigma, and there is a style like this. Now this is a hollow tube. Now what happens? The position of the anther is located this way. Alright, so the pollen from the anther, they are dropped on the stigma, and stigma being sticky easily catches the pollen, and these pollen then form pollen tube, and they release the male gamete in the style, which is a hollow tube, and then it leads to the female gamete here, and the fusion takes place and produces what is known as the seed. So this is what is happening in a self-pollinating plant. Alright. Now, in cross-pollination, what happens? We have in our environment what are known as the pollinators. Alright, like honeybee, bat, hummingbird, the nectar-sucking birds, the butterflies, etc. And majorly, pollinators are the honeybees. And in case of the mustard, honeybees are the major pollinators. Now, in case of this cross-fertilization, what happens? Now in case of cross fertilization, there is a stigma, this is the style, in one flower. Okay. Now in another flower there is anther. Air dispersal, the honeybee, the pollinators and etc., the biotic and the abiotic factor, they disperse the pollen from here, and this stigma catches the pollen. Now if the pollen are of the same variety or of the same species, because there is no cross species breeding, all right, so if it comes from the same species, from the same variety, the pollen will form the pollen tube and release the ova in this in this part, all right, and this part is known as the thalamus. Okay, and it will then fuse with the female gamete and a seed will be formed. All right, so this is what is called as cross fertilization. So far we have understood what this self fertilization means and what this cross fertilization or cross pollination basically means. So pollination is first and then comes the fertilization after the male and female gametes they fuses. So this is what the process is happening. So we have understood the process now. So I am hopeful that it will be easy for you to understand that how this DMH-11 mustard has been formed and what is this Buster-Barnett system. So we have seen the diagram. We've talked about. All right. Now let's move on. Now how this hybridization was achieved. Now basically, when we talk about the BT cotton, it has the BT name is derived from a soil bacteria called as Bacillus thuringiensis. Now this Bacillus thuringiensis has a gene called cry and it produces a chemical compound which is lethal for the bollworms, and that is the reason why the BT cotton survives the attack of bollworm. Now in case of this DMH mustard, the natural variety is not resistant to the herbicides or the weedicides which are sprayed. This hybridized variety is resistant to the herbicides, is resistant to the weedicide. Now how this has been achieved? By manipulation. All right. The Delhi University's Center for Genetic Manipulation of Crop Plants had developed this hybrid mustard DMH-11. All right. Now it consists of two different alien genes from a soil bacteria called Bacillus amyloleucophaciens. All right, in BT cotton it was Bacillus thuringiensis. All right, in here it has been, the alien genes have been taken from the soil bacteria. Now first happens the first gene, which is known as the Barnes genes. This Barnes genes, it codes for a protein which impairs the pollen production and it induces what is known as the male sterility. Now you're looking, a self-pollinating plant is being turned into a plant with only stigma, because if pollen are impaired, the anther is no longer functional. The only thing that is functional in a flower will be the stigma, will be the female reproductive organ. The male reproductive organ, which is constituted of this anther and this stamen, is dysfunctional now. The only functional part is the female reproductive system. All right. So this Barnes, it codes for a protein which induces male sterility, but female reproductive portion is still intact. Now what will happen? Now this plant is now then will be cross pollinated with the fertile parental line. All right. So क्या किया गया यहाँ पे? एक प्लांट को लिया, उसमें मेल स्टेरिलिटी इंड्यूस कर दी. Now the another, it was mate with another plant from the parental line. अब parental line से ही एक दूसरा plant लिया. अब now what will happen? We have to stop the functionality of this Barnes genes also. If Barnes genes is going to continue, then it will have an effect on this also. All right. So what will happen? Now the second gene will come into picture, which is the bar star. Now this bar star gene it blocks the action of the Barnes gene. Now male sterility is removed. Therefore, a plant is there having a female reproductive system and a plant is there which is having a male reproductive system. Now cross fertilization or cross pollination is achieved this way. Now what we will have as a progeny will be the hybrid variety, the first filial generation. Alright, so therefore the first filial progeny is both high yielding and it is capable of producing seeds and grains. Alright, so according to this Center for Genetic Manipulation of Delhi University, this technique is robust to hybrid mustard as well as the seed, and it can be used for n number of varieties. For example, the Indian mustard variety by the name Varuna and the East European early Hera 2 mutant. Now both these can be used to both these varieties can be used to develop hybrid using the Barnes-Buster technique. Alright, so I am hopeful that so far you understand what this genetic modification is. So genetic modification basically implies the modification in genes. Modification in genes. Now what are these genes? What are these genes? You see, this is our cell. And in cell we have the nucleus, and in nucleus we have our genetic material, which is DNA, and sometimes it can be RNA also when it comes to the virus. DNA virus are also there, RNA viruses are also there, which are known as the retrovirus. For example, the virus for the cold, the common cold is a retrovirus. Alright, so genetic material is there. Now this genetic material is manipulated. Either some part of it is cut and some other gene is introduced, like in case of the Bt cotton, the Bacillus thuringiensis cry gene was introduced, so that when the gene is expressed, the cry protein will be expressed, which is basically lethal for the bollworm, right? So this is what the genetic modification is. So and the hybrid mustard is also a GM crop. So genetic modification understood? Alright, let's move on now. So let's see the role of GEAC. Now basically it's an appraisal body which is responsible for the release of the GM crop, as we discussed in the introduction also. What it has done, it has recommended the seed production and the field testing of the DMH-11 Mustard variety. And it has also recommended the release of the parental for them to be used for the production of the hybrids. Now generally what happens in case of the Bt cotton, the seed production is very low or negligible. The seed production is very negligible. So therefore a farmer has to buy a new batch of seeds in order to have GM Bt cotton, in order to grow Bt cotton. Every time a farmer grows Bt cotton, a fresh batch of Bt cotton seeds are bought by the farmers. However, in case of the hybrid mustard variety, the seed production can be introduced as per the Center for Genetic Manipulation. So seed production can be enhanced. Therefore, the farmers will not have to buy the seeds every time it has to grow this crop. Alright. The significance is this: basically in India the genetic variety of the mustard is very few. And this system, the Barnes-Buster system, is going to help us develop new varieties, introduce new traits, introduce more climate smart agriculture, and climate resilient agriculture, to have the varieties which are resistant to the climate change. So therefore this system becomes very important. This is the significance of releasing and having the commercial and the field trials of the DMH mustard, and also pest and disease resistant varieties like I talked about. For example, using varieties like Don's, Kaja and Hera. These are two different varieties of the mustard. Why GEC took so long? Like I said, there were opposition from the health and the environmental activists. Like genetic modification, we don't know what we are going to produce. What if it is toxic? What if it goes into the body and produces toxicity? So we have no idea. Even if it gets successful in the field trials, even if it gets successful in producing the seeds to the particular level of replacement level of the seeds which is required by the farmers, what if it turns out to be toxic in the environment? What if it turns out to be toxic when one ingests it after the metabolism? So there are certain health concerns with respect to this, and also the presence of the third gene, which is known as the BAR gene, which makes this GM mustard plants tolerant to the weedicide. tolerant to the weedicide called glufosinate. So we are basically developing a highly resistant variety. We are basically developing a highly resistant variety. So which the implication of which are not known, the future incidents are not known. Therefore, there are certain concerns raised by the health and environmental activists that we do not know what it is going to produce once it is introduced in the environment. And talking about the resistance, BT cotton in recent years have been attacked by the bollworms, because bollworms have developed resistance against the BT cotton gene. All right, so persistent and resistant is not ensured, like we saw in the cases of BT cotton. And also, it is considered by the farmers that the big companies, they monopolize the genetically modified crops. In BT cotton it happened. The Monsanto firm, which is the largest producer of the BT cotton seeds, has basically monopolized the market. The whole market has been monopolized, and the cost of the seeds are very high, due to which the farmers has to suffer. Therefore, there have been a persistent resistance by the environmental and the health activists, and also some farmers too. According to the scientists, now scientists are definitely, they are giving the explanation that the bar gene is only a marker gene. Now what is this marker gene? Now tell me, in plant variety, how would you recognize that which is hybrid and which is not hybrid? There has to be a certain trait which help you identify that this is my hybrid variety now. So bar gene basically works as the marker gene to help the scientist and to help the people identify that this one is the hybrid variety now. A certain more identifiable trait is chosen as a marker gene so as to identify phenotypically by appearance. Just by appearance we are unable to identify that this one is the hybrid variety. All right. So far we have seen that another concern which is raised by the environmental activists is about the pollinators. Climate change has a very bad impact on the pollinators. The consistent change in the magnetic field of the earth and the climate change is having an adverse effect on the environment, pollinators, especially the honeybees. Therefore, GEC has recommended, in conclusion, basically GEC has recommended that field studies on pollinators too need to be conducted so as to understand the whole ecosystem process, so as to understand the whole ecological cycle which is going to be played since the time of cropping until the time it is harvested. In conclusion, India has a high demand of edible oil. We import approximately 14 to 14.5 million tons of edible oil, including the oils of sunflower oil, palm oil, mustard oil. All right. And in India, we have less than 6 tons of production, approximately 3 to 4 tons of million tons of production, which is very less as compared to the domestic consumption and the domestic demand. In order to reduce the import bill, in order to reduce the foreign exchange flow, the genetically modified varieties are important. So this is how we are going to conclude it. All right. So that's it for today. Thank you so much for watching Tarun IAS. Have a nice day.